Have you ever really stopped to think, really think how a vaccine works? Beyond just it stops you getting sick. What's actually going on deep inside your body?
It's fascinating, isn't it? It's not just, a simple shot. It's more like this really intricate conversation starting up between your immune system and, well, basically a most wanted poster we show it.
A wanted poster, I like that.
Yeah. And today, we're going to dive deep into that whole world. It's complex, sure, but truly amazing. We want to pull back the curtain on the immunology of vaccination.
Absolutely. And for this deep dive, we're drawing heavily from a really comprehensive lecture. It's called Immunology of Vaccination, given by Joshua Leighton back in October 2021. Think of this as maybe your express lane to getting a real handle on this vital stuff without wading through dense textbooks.
Right. Our goal today, really, is to pinpoint those key mechanisms, the immunological gears that make vaccines work. We'll meet the cellular cast, figure out the different kinds of immune responses, look at the various ways we vaccinate, and importantly, touch on some of the hurdles vaccine developers face.
Hopefully providing you with some real aha moments along the way.
Exactly, connecting the dots for a solid understanding.
So this whole intricate dance,
Yeah.
how does it even begin? At its core, vaccination is all about preparation, isn't it?
It really is. Think of your immune system as this highly trained security force, always patrolling. Vaccines are like their specific training drills. They show the force what a potential intruder looks like before any real invasion happens.
So when the actual bug shows up,
Yeah.
your body's defenses are already prepped and ready. They know the playbook.
Precisely. And to really get this, we need a few key terms under our belt. They're central to this whole immunity story. First up, antigen.
Okay, antigen. That's like the unique identifier on the intruder, the thing the immune system spots as foreign.
You got it. And then on that antigen, there's an even tinier, more specific spot called an epitope.
Ah, the epitope. So that's the precise bit the antibodies actually latch onto, like a specific handhold.
Exactly like a handhold or a key fitting a lock. Perfect. Then there's immunogenicity.
Immunogenicity. Sounds complex.
It just means how good something is at, provoking that immune response, making the immune system sit up and pay attention. Some things are just better at waving that red flag.
Gotcha. And, if I had to pick one defining feature, one superpower, it's got to be immune memory, the ability to remember past encounters.
Oh, absolutely crucial. It's the whole basis for long-term protection from vaccines. It's like your immune system keeps a detailed file on every threat it's ever dealt with.
And sometimes when that system is learning, you feel it, right?
Right.
That's reactogenicity.
That's it. Those mild side effects, sore arm, maybe a bit of fever, it's actually often a good sign. It means your immune system is engaged, it's learning, building those defenses. It's the system doing its job.
Okay. And the final term for now, seroconversion.
Seroconversion. That's basically the point where we can actually measure the antibodies in your blood. It shows your body has responded either to an infection or a vaccine and is now, armed and ready.
So we've got the terms. Now, who are the actual players? The cells doing all this work?
Right, the immune orchestra. It's amazing, really.
Yep.
All these incredibly diverse cells, they all start from one single source, the hematopoietic stem cell, a master cell.
One starting point for everything. Wow.
Yep. And from there, they go down two main paths. One leads to the myeloid progenitor cells.
And those become?
Those become your rapid responders. Think mast cells, eosinophils, basophils, monocytes, neutrophils, plus the crucial communicators, dendritic cells and macrophages. They're often the first ones on the scene.
First responders.
Okay.
And the other path.
That's a lymphoid progenitor path. This gives rise to the real specialists. T cells, B cells, NK cells, and plasma cells. These are the ones involved in that highly specific adaptive response we talked about.
T cells and B cells, those names come up a lot with vaccines.
They're central players, absolutely.
Okay, so we have this whole roster of cells. How do they coordinate? How do they know what to do? This leads us to the two main branches of defense, right? Innate and adaptive.
Exactly. Think of innate immunity as your body's immediate reaction force. It kicks in super fast minutes, hours maybe.
Quick off the mark.
Very quick. But it's not specific. It recognizes general danger patterns, these things called PMPs, pathogen-associated molecular patterns, basically common molecular signatures found on many microbes.
So it sees broad categories of trouble, not necessarily specific identities.
Precisely. And crucially, innate immunity doesn't create long-term memory. Your macrophages, neutrophils, NK cells, dendritic cells, they're key players here, trying to contain things immediately.
Okay, that's the innate side. Fast, general, no memory. What about adaptive?
Adaptive immunity is the slower, more deliberate branch. Takes days, maybe weeks to fully develop.
Slower, but I'm guessing more powerful in the long run.
Definitely more precise. It's highly specific to particular pathogens, even specific epitopes on antigens. It learns to distinguish self from non-self with incredible accuracy. And this is the branch with that all-important immunological memory.
And this is where the T cells and B cells, those lymphoid cells, really take center stage.
Absolutely. They're the stars of the adaptive show, the strategists, the memory keepers.
Got it. So we've laid the groundwork, the cells, the basic concepts, the two arms of immunity. Now let's really dig into that adaptive system. The humoral and cell-mediated responses sound critical. Where do antibodies fit in?
Right, so let's zoom in on adaptive immunity. When people think immunity, they often think antibodies, those Y-shaped proteins. And the cells responsible, that's primarily the B cells. They are the heart of what we call the humoral response.
Humoral, meaning related to the body fluids, like blood.
Exactly, where antibodies circulate. When a B cell gets activated by its specific antigen, it undergoes this incredible transformation. It becomes a plasma cell.
And plasma cells are.
Think of them as dedicated antibody factories, just churning out massive quantities of these specific antibodies designed to target the invader.
Wow. I understand there isn't just one type of antibody, right? There are different classes.
That's right. Different classes or isotypes of immunoglobulins, each with slightly different jobs. First up is IgM.
IgM, what's its role?
IgM is usually the first antibody type produced when you encounter a new antigen or pathogen. It's like the first wave of defense. In the blood, it actually exists as this large pentamer structure, 5 units joined together.
Like a big grappling hook.
Kind of. It's very good at grabbing onto things and activating other parts of the immune system, like the complement cascade, which helps destroy pathogens. It's also found as a monomer, a single unit, on the surface of naive B cells waiting to be activated.
Okay, first responder IgM. Then what?
Then comes IgG. This is the most abundant antibody in your blood serum. It's the real workhorse for long-term protection.
And IgG has a unique trick, doesn't it?
It does. It's the only antibody class that can cross the placenta from mother to baby.
That's incredible. So mom's immunity gets passed on directly.
Exactly. Provides vital protection for the newborn. IgG is also great at activating complement and neutralizing toxins. There are actually 4 subclasses, IgG1, 2, 3, and 4, with slightly different functions. Many vaccines aim to generate lots of high-affinity IgG.
Makes sense. What about IgA?
IgA is your mucosal defender. It's the main antibody found in secretions. Think saliva, tears, mucus in your respiratory and gut linings.
So it's guarding the entry points.
Precisely. It forms a dimer, 2 units joined together, and provides critical defense against bacteria, fungi, and viruses right there at the mucosal surfaces. It's our frontline shield in many ways.
Okay, IgM, IgG, IgA. What's next?
IgD.
IgD. It's a bit of a mysterious one. Like IgM, it's found on the surface of naive B cells, but there's much less of it in the blood. Its exact function is still, well, debated. Some evidence suggests it might play a role in allergic responses or B-cell activation, but it's not as well defined as the others.
Still some secrets the immune system holds. And finally, IgE. IgE is the least common in serum, but it punches above its weight. It's famously involved in allergic reactions. It binds to receptors on mast cells and basophils, triggering histamine release.
Ah, the allergy antibody.
Right. But it also plays a really important role in defending against parasitic infections like worms.
So different antibodies for different jobs. And vaccines are often designed to trigger specific ones, like IgG.
Exactly. Getting the body to produce high levels of protective antibodies, particularly long-lasting IgG and mucosal IgA, is a primary goal for many vaccines. That's the essence of the humoral response target.
Okay, that covers the antibody side.
Yeah.
What about the other arm of adaptive immunity, the cell-mediated response? You mentioned T cells earlier.
Right. So cell-mediated immunity is driven by T cells, and they operate differently from B cells. B cells can often recognize antigens directly, but T cells need help. They only recognize protein antigens, and crucially, these antigens have to be presented to them.
Presented? How does that work?
By other cells, usually antigen-presenting cells like dendritic cells or macrophages or even infected body cells. These cells chop up the antigen internally and display little fragments on their surface using special molecules called MHC molecules, major histocompatibility complex.
So MHC molecules are like little display stands.
That's a perfect analogy. They hold up the antigen fragment for the T cell to inspect. There are two main classes, MHC class I and class II, which interact with different types of T cells.
Okay, and what are the main types of T cells doing this inspecting?
Two major players here. First, the CD8 plus T cells, often called cytotoxic T lymphocytes, or CTLs.
Cytotoxic sounds serious.
They are. Their main job is to find and kill body cells that are infected with viruses or have become cancerous. They recognize antigen fragments presented on MHC class I molecules, which are found on almost all our cells. They're the assassins, eliminating threats from within.
Direct killing. Got it. And the other type?
The CD4 plus T cells, or helper T cells, these are the conductors of the immune orchestra.
Helper cells, what do they help with?
Pretty much everything. They recognize antigens presented on MHC class II molecules, which are mainly found on antigen-presenting cells. When activated, CD4 plus T cells don't usually kill cells directly. Instead, they secrete chemical messengers called cytokines.
Cytokines, chemical signals.
Exactly. These cytokines guide the entire immune response. They help activate B cells to make antibodies. They boost the activity of CD8 plus T cells. They recruit other immune cells to the site of infection. They coordinate the whole effort.
So CD8 plus T cells are the killers, CD4 plus T cells are the coordinators, both vital.
Absolutely essential for a well-rounded adaptive immune response, especially against complex pathogens like viruses.
And all this ties back to that idea of memory, right? The primary versus secondary response.
Precisely. That first time your adaptive immune system sees a new antigen, maybe from a vaccine or a first infection, that's the primary response. There's a lag phase while the right B and T cells are found and activated, then antibody levels rise slowly, mostly IgM first, then IgG. It works, but it takes time.
Okay, the first encounter is a learning process.
Right. But because of immune memory, those long-lived memory B cells and memory T cells created during the primary response the second time you encounter that same antigen, the secondary response kicks in.
And it's different how?
It's much faster, much stronger, and lasts longer. The lag phase is shorter, you get way more antibodies produced, much quicker, and it's predominantly high-affinity IgG. The memory cells are already there, primed and ready to go.
So that's why vaccines are so effective. They establish that memory, so if you meet the real pathogen later, you get that supercharged secondary response immediately.
That's the core principle, exactly. You train the system without causing the disease, so it's ready for the real fight.
This naturally leads us to think about the different ways we actually achieve immunity. You mentioned active and passive immunity earlier.
Yes, two fundamental ways your body can gain protection. Let's start with passive immunity. Think of this as borrowed immunity. It's temporary.
Borrowed. Like how?
The classic example is a mother passing antibodies to her baby. This happens across the placenta during pregnancy, mainly IgG, and also through breast milk, which is rich in IgA.
So the baby gets immediate protection from mom's antibodies.
Exactly. It's crucial for those first vulnerable months, but it doesn't last forever because the baby isn't making those antibodies itself. They eventually degrade. Another example is giving someone an injection of preformed antibodies, like IVI treatment for certain conditions or exposures. Quick protection, but temporary.
Okay, so passive is borrowed and temporary. What's active immunity?
Active immunity is when your own immune system does the work. It encounters an antigen and mounts its own response, developing those all-important memory cells. This provides long-lasting, sometimes lifelong protection.
And how do you get active immunity?
Two main ways. Naturally, by getting infected with a pathogen and recovering, your body fights it off and remembers it. Or, the way we prefer, through vaccination.
Right. Vaccination triggers your own immune system to build that active, lasting memory without you having to go through the illness itself.
Precisely. It's a safer, controlled way to achieve robust, active immunity.
Now, you mentioned earlier that different types of vaccines can actually trigger slightly different immune responses. That sounds really important for vaccine design.
It's incredibly important. Let's look at a few main types. First, live attenuated vaccines, or LAVs.
Live attenuated, meaning weakened.
Exactly. These use a version of the actual living virus or bacterium that has been weakened or attenuated, so it can't cause significant disease in healthy people, but it can still replicate a little bit in the body.
So it mimics a natural infection quite closely.
Very closely. And because of that, LAVs tend to induce a really broad and strong immune response. You get excellent memory CD4 plus T cells, importantly, memory CD8 plus T cells, because the virus replicates inside cells, high affinity IgG and often IgA, and strong B cell memory. Think MMR or chickenpox vaccines.
Robust response covers lots of bases. Okay, what about inactivated vaccines?
These use the virus or a bacterium, but it's been killed, usually by heat or chemicals. It can't replicate at all.
It's safer, perhaps? But does it trigger as strong a response?
Generally, the response is still very good, but might be slightly different. They induce good memory CD4 plus T cells, high affinity IgA, and B cell memory. But because there's no replication inside cells, they typically don't induce as strong a CD8 plus T cell response as LAVs might. Examples include the inactivated polio vaccine or some flu shots.
Okay, still good memory, but maybe less emphasis on the cell-killing T cells. Next.
Subunit vaccines. These don't use the whole pathogen at all. Instead, they use just one or more specific pieces, or subunits, usually proteins or sometimes sugars from the pathogen surface.
Like just showing the immune system that wanted poster fragment we talked about.
Exactly. Think hepatitis B or a cellular pertussis vaccines. They also induce memory CD4 plus T cells, high affinity IgA, and B cell memory. But there's a key thing about subunit vaccines.
What's that?
They often require adjuvants.
Adjuvants. Those are substances added to vaccines, right?
What do they do? Well, because subunit vaccines are just purified components, they often lack those general danger signals, the PMPs, that the innate immune system recognizes so well from whole pathogens like in LAVs.
Ah, so they might be too clean to trigger a strong initial alarm.
Precisely. Adjuvants act as that alarm signal. They help stimulate the innate immune response, basically telling the immune system, hey, pay attention to this protein. This helps ensure a much stronger and more durable adaptive response, enhancing antibody production and memory development. They give the vaccine that necessary kick.
Got it. Adjuvants provide the danger signal that the purified subunit lacks. Makes sense. Are there other types?
One more major category is polysaccharide vaccines. These use the long chains of sugar molecules, the polysaccharides, that make up the outer capsule of certain bacteria like pneumococcus or meningococcus.
Just the sugar coating.
Yep. Now, these are interesting. They primarily stimulate B cells directly, often without much T cell help. This tends to induce mostly lower affinity IgG and IgM antibodies.
Lower affinity and memory.
And typically, no significant B cell memory is generated. The response can be protective, especially in adults, but it's often not as long-lasting, and it doesn't work well in young infants whose immune systems don't respond well to polysaccharides alone.
So less durable protection, especially for kids. Is there a workaround?
Yes, that's where conjugate vaccines come in. They chemically link the polysaccharide to a protein carrier. This clever trick engages T cell help, leading to a much better response, higher affinity antibodies, class switching to IgG, and importantly, immune memory, even in infants. The High vaccine is a classic example.
Clever. Linking the sugar to a protein makes it act more like a protein antigen, engaging T cells.
You've got it. It dramatically improves the vaccine's effectiveness and duration of protection.
Okay, so different platforms, different responses, different strategies like adjuvants and conjugation. It's quite complex. This must lead to some real challenges when developing vaccines, especially for tricky pathogens.
Absolutely. And the immune system itself faces different challenges depending on what it's fighting. Let's compare viral and bacterial immunity.
How do they differ?
With viral immunity, a big focus is dealing with the fact that viruses replicate inside our own cells. So yes, you need antibodies to neutralize viruses in the blood and fluids before they infect cells.
Stop them at the door.
Right. But once they're inside, you need ways to eliminate those infected cells. That's where those CD8 plus cytotoxic T cells, the CTLs, are critical. NK cells also help. The goal is to clear the virus from the body and establish memory to prevent reinfection.
So both antibody and cell-mediated arms are crucial for viruses. What about bacteria?
For bacterial immunity, antibodies are also vital for clearing bacteria from blood and fluids, a process called opsonization, where antibodies coat bacteria, making them easier for phagocytes to eat. And yes, if bacteria get inside cells, CTLs and NK cells can be involved too.
Similar so far.
But a major difference is that many harmful bacteria cause disease by producing toxins. So a huge part of bacterial immunity is neutralizing these toxins, often with antibodies. Think tetanus or diphtheria. The vaccines primarily target the toxins, not the bacteria themselves. And again, immunity to reinfection is key.
So toxin neutralization is a big factor for bacterial defense. And pathogens themselves can fight back, right? Make it hard to develop vaccines.
Oh, definitely. Microbes are incredibly resourceful. They have numerous evasion strategies. The lecture mentioned the parasite Trypanosoma brucei, which causes African sleeping sickness.
What does it do?
It employs something called antigenic variation. It has a vast repertoire of genes for its surface coat protein. Periodically, it just switches to expressing a different coat protein.
Like changing its clothes constantly.
Exactly. Just as the immune system mounts a strong antibody response against one coat, poof, the parasite population switches to a new coat that those antibodies don't recognize. The immune system has to start all over again.
Wow, that's tedious. So the parasite stays one step ahead.
It leads to those fluctuating levels of parasites in the blood seen in the disease, and it makes developing a vaccine incredibly challenging. What do you target if the target keeps changing? It's a major hurdle for diseases like malaria and sleeping sickness.
A constantly moving target. That really highlights the challenge. And there was another nuance in differences between individuals.
Yes, and this is a really important and increasingly studied area. The lecture highlighted that vaccine-induced immune responses can differ between biological males and females.
Really? How so?
The mechanisms are complex and still being fully understood, involving hormonal influences, genetic factors, even the microbiome. But generally, females often mount stronger innate and adaptive immune responses, including higher antibody production after vaccination compared to males.
Does that mean vaccines work better in females?
Not necessarily better, but potentially differently. It might mean females sometimes experience more reactogenicity, those side effects, because their immune system is reacting more strongly. It also raises questions about whether optimal vaccine doses or schedules might eventually need to be tailored.
That's fascinating. It adds another layer of complexity to the one-size-fits-all approach.
It really does. It points towards a future where vaccinology might become more personalized, taking these biological variables into account to maximize effectiveness and safety for everyone.
So reflecting on everything we've covered, it's just, well, staggering. The sheer complexity, the elegance of the immune system, and how vaccines cleverly tap into all these natural processes.
It really is remarkable. From the initial danger signals and innate response, through the intricate activation of specific T and B cells, the production of tailored antibodies, and the establishment of that crucial long-term memory. It's a biological masterpiece.
And understanding even the basics, like we've discussed today, really gives you a much deeper appreciation for what's happening when you get a vaccine. It's not magic. It's incredibly sophisticated science, harnessing your body's own defenses.
Absolutely. It underscores the power of these public health tools and the ongoing scientific effort to refine them and tackle new challenges. It's a constant evolution on both sides, pathogens evolving and our understanding and interventions evolving too.
Which brings us to a final thought to leave you with. Considering all these nuances we've touched on, the different responses to various vaccine types, the challenges posed by evasive microbes like trypanosoma, and these inherent differences in immune responses, like those between biological sexes, what does this complexity really suggest about the future? Where might personalized vaccinology take us, and how will we continue our vital, ongoing fight against infectious diseases in an ever-changing world?